So we have a pretty good feel for what double-stranded DNA looks like. Of course, the chromosomes have enormously long stretches of double-stranded DNA. Some of the chromosomes are about 100 million nucleotides in length. There's some that are 250 million nucleotides in length, so they're incredibly long molecules. But that's not the way they exist in the nucleus. They exist as chromatin, what's called chromatin. They're complex with proteins. So it's not naked DNA in the nucleus, it's actually complex with proteins which are called histones. And that's what we're going to be looking at today. And they're arranged, the subunits of how these things are packaged are called the nucleosomes. We're going to look at those as well. And if you look at the picture on the right-hand side, it shows the nucleus in the top left corner, and then these nuclei were lysed gently to allow the DNA to spill out. And you can see how much, and this is just a very small portion, you can see how much chromatin, how much DNA exists in the nucleus. It's literally jam-packed with DNA. And so the nucleosomes and the histones do a couple of things. One is they protect the DNA, and we'll see later they're involved in gene expression. But they also compact the DNA so that you can, and they compact it in a very organized fashion so that you can get all the chromatin, all the material, all the DNA into that nucleus in a way that's organized, which makes it accessible for gene expression and DNA replication. It's not just a big mess in there. It's not just stuffed in there. It's highly, highly organized. So let's start taking a look at what chromosome composition or what chromatin actually looks like. So again, we're looking at chromosome composition, and again, the DNA is complex with histone proteins, and that's what we call chromatin. So the material in the nucleus is referred to as chromatin. Highly organized, highly compacted, and the basic structural unit is the nucleosome. And you can see the nucleosome under electron microscopy. So I'm showing you two images here. The one on top is an image where the cells were gently lysed with a very weak detergent, a mild detergent, and the chromatin comes spilling out of the nucleus, and then some of that was then put on a grid and then looked at under the electron microscope, and what you can see is that it looks like a thick fiber which is made up of lots of little dots. Each one of those dots is a nucleosome, and this fiber here is referred to as the 30 nanometer fiber because it's 30 nanometers in diameter. Now if you do the same experiment, you lyse the cells and you treat it with a mild detergent, and you let that come out, and then you treat it with a little more rigorous detergent, maybe even a little bit of heat, and then you look at the chromatin then under the microscope, you see what we see at the bottom. So this is referred to as decondensed chromatin. The 30 nanometer fiber is condensed chromatin, and after treating with the detergent, you get what's called the decondensed chromatin, and what you can see is now those individual dots, those individual clusters. So you have a cluster, a space, cluster, a space, cluster, a space. We'll look at a drawing in the next diagram, but these are nucleosomes. This is, sometimes you hear this is referred to as the bead on a string, because there are beads with almost like a necklace, beads on a string. So let's take a look at the nucleosome, because the nucleosome is clearly the primary subunit of the chromatin. So we're looking at the chromatin composition, and we're asking the question, what is the nucleosome? So I have a drawing here which represents the decondensed chromatin, which is what we saw in that last figure. And so you see the nucleosomes are on the DNA, And then between each nucleosome is referred to as linker DNA. That's 80 to 100 base pairs of literal double-stranded naked DNA. So you have nucleosome, naked DNA, nucleosome DNA, and so on. And they're separated by about 180 to 100 base pairs. Now the question that I want to address is, what is this nucleosome? So we have ways of trying to analyze it, trying to determine, is it protein, is it nucleic acid, is it RNA, lipid, what is this stuff? So here's how an experiment is done. We'll break it up into a few parts, but first of all, isolate the chromatin with detergent so you get that bead on a string form. Get a lot of it in a test tube. Treat mildly with DNase. DNase degrades the DNA, so what it's going to do is degrade the linker DNA between each nucleosome. And then what you have in the tube then is free nucleosomes with none of the linker DNA. So now you have isolated, basically purified nucleosomes. And then what we do is we take those nucleosomes and we denature them. We break them apart. Usually use high salt and heat. You can boil it, breaks it all apart, and then you assay or analyze for what's in there. Is it proteins? Is it DNA? Is it both? And the analysis is done by gel electrophoresis, which is a very important technique. It's a very simple technique, but it's used all the time in molecular biology. And so I'm going to describe to you, some of you may have done it, some of you may be familiar with it, but most people may not be, so I'm going to describe what's called gel electrophoresis. So here we have a figure which pretty much describes what this gel electrophoresis does. The gel electrophoresis is used to analyze DNA, protein, RNA, and basically to determine the size of the molecule. So if you look at the left-hand side, it's kind of bluish. We have what's called a gel slab. This is considered a semi-solid medium. It's kind of like Jello. It's agarose, usually 0.5% or 1%. But this is a semi-solid media, meaning that it's very liquidy, and material can pass through it. So it's not so solid that material cannot migrate through it. And then you see at the top, those white little rectangles, those are wells in the gel where you can actually place material. Now, the two ends of the gel are connected to liquid, which is basically an electrolyte solution. Could be sodium chloride, something that will carry a charge. And they're connected, here they're showing it connected with a kind of a wick that's wet. And then you hook that up to a power source, which is shown there, and you turn on the power source and you generate a voltage. And the voltage runs through the gel from in this case, from the top, which is on the right, which is the negative electrode, to the positive electrode. So the charges run towards the positive electrode. Now, if you place your samples in there, and you can place protein, you can place DNA, you can place RNA, you can place a mixture. So if you place your sample in the wells, and then you turn that on, these samples, the DNA or protein, will migrate through the gel as a function of its size, because it has to make its way through this gel. So small molecules, small pieces of DNA will run very quickly because they can make it through the gel much easier. Very large molecules will move more slowly through the gel, and they move in a uniform way. So what it shows on the right-hand side is they're looking at DNA samples. So in the first figure, the samples on the left, the samples are loaded. So you have different samples, different mixtures of DNA loaded into each well. You turn the power on, and they migrate through the gel. And the ones that are small migrate more quickly, and the ones that are large migrate more slowly. So you can see how they separate into different bands because the group will migrate together, whatever, according to its size. So again, this is used to analyze DNA, protein, or RNA, and different stains will stain different materials. So something called ethidium bromide is a stain that stains DNA and RNA. What it is it's called an intercalating agent. It fits in between the base pairs, but it's also fluorescent. So if you put this material in the gel, it'll bind to the DNA, and then if you shine a UV light on that, you'll see the fluorescence of the DNA or RNA. And Kamasi blue is another stain, but that specifically stains proteins. Binds to the proteins, and you can see it as blue. So the bands will be blue. So that's the gel electrophoresis. And so what happened was we took the nucleosomes that were purified, broke them up with heat and all kinds of other reagents, and then prepared these gels, and then separated the components on the gels. And this is pretty much what they found on the next slide. So here on the right-hand side, it shows, again, the procedure. The beads on the string form a chromatin, decondensed chromatin, isolated, digested with nuclease. And then it says dissociation with high salt and maybe some heat. And everything comes apart. And then you dissociate everything, and then you run it on these gels. One gel is stained with ethidium bromide. That stains the DNA, and the other was stained with Kamasi blue. So if you look on the left-hand side, you see the protein gel, and what we see are 4 distinct bands. Only 4 bands. So the nucleosomes contain 4 protein species, different sizes. And they're referred to as H2A, is the biggest one, H2B, H3, H4. And by calculating the stoichiometry, it turns out that for each nucleosome, there's 2 copies of each. So 2 copies of H4, 2 copies of H3, and so on. So there's actually 8 proteins in each nucleosome. And then and that's referred to as the histone octamer, 8 proteins. And then if you look at the DNA stain gel, what you see is a single band of 146 base pairs. So the nucleosome contains 8 histone proteins, the histone octamer, and 146 base pairs. So first I want to tell you a little bit about what the histones look like. So there are these 4 histone proteins. I have them kind of illustrated here, H2A, H2B, H3, and H4. They're pretty tiny proteins. The biggest one is H2A, which is 140 amino acids. H4 is 102 amino acids. These proteins are highly conserved. I think, as far as I recall, they are the most highly conserved proteins that we know of. If you look at plant histones, or if you look at mammalian histones, there's very few changes, maybe 1 or 2 amino acid changes, but they're very, very highly conserved. Now, the interesting thing about them is that they are highly enriched in lysine and arginine. And if you look, I have on that table there, I have the percent lysine arginine, and so the H2A, for example, well, H2A is 20%, H2B is 22%, H3 is 25% lysine and arginine. 25% lysine and arginine, which is an enormously high proportion of these 2 amino acids. So, for example, in the histone H3, which is 25% lysine arginine, there's about 30 lysine and arginines in the protein. Whereas if it was just random, there may be 5 or 6. So there's a very high percentage of lysine and arginine, and if you remember back when we looked at the amino acids, the lysine and arginine were basic amino acids, and that means that these are positively charged at neutral pH. So all of these histones are highly positively charged proteins. Now, the lysines and arginines are located in specific regions, and that's kind of shown below in the diagram. The lysines and arginines and these positive amino acids are generally located at either the amino terminus or the carboxy terminus at the kind of the ends of the protein. And the center of the protein is made up of more nonpolar amino acids, so it forms kind of more of a globular shape. That's what's kind of shown. It'll try to illustrate that below. So the protein kind of has a complex folding, but the two edges, the amino terminus and the carboxy terminus, are exposed to solution in a sense because they interact with the aqueous environment because of the hydrogen bonding. Now, people have done a lot of work. They've done X-ray crystallography. They've done the biochemistry, trying to sort out what the histone proteins look like in the context of the nucleosome and the DNA. And as it turns out, these histone proteins, that's two of each of these, complex together in a very specific way, and the DNA wraps around the nucleosome. So that's what's shown here, where you have the histone octamer plus 146 base pairs of DNA. And if you look at on the left-hand side, it shows the proteins, illustrates the proteins kind of self-assembling into this histone octamer. We have two copies of H2B, two of H2A, two of H3, two of H4, and then the DNA 146 wraps around that. And then you have the linker DNA, and that's another nucleosome. Now, on the right-hand side, what I'm showing you is an image generated by X-ray crystallography. And in this case, when this was done, this is an entire nucleosome with the DNA that was crystallized. And then they carried out the X-ray crystallography, and they generated the image. And then this was the image that was generated. And so you can see it kind of supports what the model is. It demonstrates it. This is the DNA and the histones co-crystallized. And so you can see the DNA on the outside. You can see the double helix. And on the inside, you can see the proteins. They're represented by different colors: orange, red, blue, and green. And if you look at the right-hand side there, that's kind of a side view, and you can see the DNA is wrapped around twice. And you can also see, in some cases, depending on how they show it, how it's turned, you can see portions of the histone proteins coming out, and those are the positively charged portions. Now, the whole idea of the positively charged histones makes sense, because DNA, we know, is highly negatively charged. It has all the phosphates, all the sugars, so it's a negatively charged molecule. The histones bind non-specifically to DNA. That means they do not bind any specific DNA sequence, but they bind all over the DNA. So they're binding to this negatively charged molecule, so it makes sense that the histones would be highly positively charged. So you have an interaction between the positive charges on the histones and the negative charges on the DNA. Now, this was an analysis of the beads on the string form, the decondensed chromatin. And it was discovered that there was a histone during the preparation, because remember, we added more of a stronger detergent, and the addition of that stronger detergent, later it was discovered, caused the release of another histone that was not present in this histone octamer. And that histone is called histone H1. And with the histone H1, there's only one copy per nucleosome. And what the histone H1 does is it binds to the DNA that circles the chromatin, and it binds to, let's say, like the ends of that. And in doing so, itself, and in addition to binding to each nucleosome, when they interact with each other, they self-assemble and they cause a higher order of coiling. And that I have illustrated here in the next slide. So here we have, if you look below at the drawing, we have a histone octamer core, the nucleosomes. Then they show the addition of the histone H1. Histone H1, one copy of the histone H1 binds to each nucleosome, and then they self-assemble in a sense. And if you look on the right-hand side, you can see that kind of circular pattern. And what that's trying to illustrate is that the histone H1s are shown in the center. They're like a lighter purple. And they self-assemble and they cause this coiling, a coiling effect with 6 nucleosomes per turn of the coil. And that is what is the 30 nanometer fiber, what I showed you at the beginning. When they gently lysed the nuclei and the chromatin came out, it looked like what's shown above. It was a thick 30 nanometer fiber with all these nucleosomes kind of attached in a very close connection, and that's due to this coiling effect that happens with the histone H1. When we treat with the higher detergent, or the stronger detergent, that histone H1 comes off. And that allows the decondensation where then you have the beads on the string. So this is the chromatin made up by the individual nucleosomes. And then in the presence of the histone H1, you get this 30 nanometer fiber, which is probably what the chromatin looks like. Now, it's even more packaged than this. This is like the second order of compaction. First order of compaction would be the winding of the DNA around the nucleosomes. The second order of compaction would be the addition of the H1 and then the coiling of the nucleosomes into the 30 nanometer fiber. So we're packaging the DNA. But in the nucleus, there's an additional compaction, and what we see are loops, long loops of chromatin. So that's this additional looping that takes place. And so this is probably more like what the DNA looks like. It's arranged in these loops. The orange line, that orange loop, it says 20 to 100,000 base pair loop domains. Those are 30 nanometer fiber. That's the chromatin that's looped. And then they are held together at the base of each loop by what are called scaffold proteins. The book calls them chromosome loop-forming clamp proteins. And they actually bind to specific DNA sequences that are in the chromatin, but they're also referred to as scaffold proteins. And then these proteins bind to a substrate, in a sense, which is called a nuclear matrix. Now, it's thought that there's some organization here, and in many cases, these loops will represent individual genes. So it's as if the genes are separated from each other physically by this kind of looping mechanism. Now, that's about all we know in terms of how the DNA is packaged. But I will say that during mitosis, so this is what happens, let's say, in an interphase nucleus, in a cell that's not not undergoing cell division. But in a nucleus that's undergoing cell division, then of course you get the condensation of the mitotic chromosomes, which is a much more sophisticated and much more complicated, and we do not understand how it happens, contraction and packaging of the DNA. And that's illustrated on the next slide. So this is showing the DNA packing occurs on several levels, and we know the first few. So you have the double-stranded DNA packaged with beads on the string, the nucleosomes, and you have with the histone H1 the chromatin fiber, the 30 nanometer fiber, and then it forms these loops like we just saw. But then and these loops are just spread out through the nucleus. But what happens is during mitosis, when there's more condensation, you end up getting that mitotic chromosome, which is shown on the bottom, then there's a whole additional level of looping that goes on, and contraction. And so it shows the mitotic chromosome on the bottom, and what it says is the net result, this is mitotic chromosomes. Each DNA molecule has been packaged into a mitotic chromosome that is 10,000-fold shorter than its original length. So there's a 10,000-fold compaction of the DNA. Then it undergoes mitosis, and the chromosomes split. You get one in each new cell, and then they decondense and they unfold, and they do it without making any knots. So there's no knots or twists or turns or kind of any kind of knotting or confusion with any other chromosomes. It's pretty amazing. Otherwise, you get all kinds of broken things and things pulling apart. Okay, so that is the nucleosomes and the packaging into chromatin. And what I want to move into next is what I call elements that are responsible for chromosome maintenance. And that's going to be specifically or regions that undergo DNA replication, and then a centromere, and then we'll also look at telomeres. So we're going to start looking at some functional elements on chromosomes. And that will come in the next PowerPoint presentation.